già scaricare dalla rete. Adesso switcho verso l'inglese per presentare il prossimo ospite. Sono Andrea Bagno. I'm from the Department of Industrial Engineering of the University of Padova. And today I have the great pleasure to introduce Professor Claudio Ronco. Professor Ronco has been full professor of nephrology by Clara Fama at the University of Padova until 2K21. He also headed the Department of Nephrology, Dialysis and Transplantation at the San Bortolo's Hospital in Vicenza, and currently is the scientific director of the International Renal Research Institute in Vicenza. I would like to say that this research institute is Professor Ronco's creature. He graduated in medicine in 1976 and specialized in nephrology 3 years later. So, Professor Ronco, we know that you are not a bioengineer by education, but we forgive you for that. And he has been the director of the laboratory of the Renal Research Institute at the Mount Sinai Beth Israel Hospital, and professor of medicine at the Albert Einstein College of Medicine in New York City. And he's professor at the University of Virginia and at the University Fudan Jiao Tong in Shanghai. He's the author of an impressive number of scientific papers, more than 1,700 papers, almost 100 books and almost 200 book chapters. And he delivered more than 1,200 plus one today lectures at international meetings. His H index is 137. So he's also the editor-in-chief of different scientific journals: Blood Purification, Contribution to Nephrology, Cardiorenal Medicine, and editor emeritus of the International Journal of Artificial Organs. He received a number of international awards because he is considered the pioneer in many areas of nephrology, including peritoneal dialysis, critical care nephrology, continuous renal replacement therapies, hemoperfusion, cardiorenal syndromes, and wearable dialysis technology. I would like to mention that he invented the first continuous renal replace therapy for neonates, the so-called Carpe Diem. It means cardiorenal pediatric dialysis emergency machine. But he also engineered many other devices for hemoperfusion and dialysis. Finally, I would like to remember that Professor Ronco has, let's say, a rather eclectic personality. When he's not working in the hospital or just around the world for his scientific engagements, it can be found on the ice hockey field in Asiago, in concert on stage with Nomadi for charity, or writing some new book. So please, Professor Ronco, the floor is yours for your keynote lecture entitled Engineering the Artificial Kidney, Memories of a Translational Research Journey and Future Challenges. Thank you for your presence.
Okay, much better. I must comment that when they present you and they say you are a pioneer, it means only one thing: you're old. Anyway, these are my disclosures, which is important to mention. It's industries for which I'm consultant. And I have some special disclosure. First of all, I'm a physician, not an engineer. And I have a different perspective, and I hope to bring to you a different perspective today. I'm a specialist in nephrology and artificial organ support, and therefore I will use this area of extracorporeal blood purification as an example of interaction between engineering and medicine. And also, I have a personal vision on how a bioengineer should be. And therefore, it might be a biased opinion, but this is what I will present to you. First of all, I ask myself, what is bioengineering? I joking, saying often to Andrea that to me, the bioengineer does not exist. At least when it leaves the university studies, afterwards probably it becomes a bioengineer, but not at the beginning. Anyway, today we're modern, so I asked the ChatGPT, the generative pre-trained transformer chatbot, and the answer was bioengineering, also known as biomedical engineering or biotechnology engineering, is a field that combines principles of biology and engineering to develop new technologies aiming at improving human health and quality of life. More than that, encompass a wide range of applications, including medical device, biomaterial, tissue engineering, computational modeling of biological systems. And also bioengineering draws on a range of disciplines, including biology, chemistry, physics, mathematics, computer science, and engineering. So bioengineers work in a variety of environments, including academic research labs, medical device companies, hospitals, and government agencies. So bioengineering has a real spectrum of application that go from biomaterials to mechanical engineering, to chemical engineering, computer science, electrical engineering, and other things including biological and neurobiology application, chemistry and biochemistry, mathematics and statistics, physics. All these things include a kind of an interaction with the medical community in order to develop what? Patient outcome that may improve day by day. Now, as I mentioned to you, I will focus mainly on my interest, which is artificial organ and extracorporeal organ support. In this slide, you see that basically we have different organs that can be supported by different devices. And what do organs have in common? Blood and the endothelium. And so basically, when I use an extracorporeal circuit, just by modifying the device that I use that goes from extracorporeal membrane oxygenation to plasma filtration absorption, you may achieve organ support in different ways. And this leads to discussion with engineers. For example, the different phases of hydraulics that, as you can see here, go from the flow in the body in artificial circuit, in artificial organs, going from the theory of fluid flow in conduits down to microfluidics. This is bioengineering. And it goes in our field from design of dialysis circuitry, which is very complex, to dialyzer manufacturing. And this is an example of interaction. We studied the flow through hollow fiber dialyzer, showing that the flow pattern optimization can be achieved by designing specific flow distributor at the entrance of the device. But we also analyzed the maldistribution of dialysis flow. You see, it is only external to the fiber, realizing a blood to dialysis flow mismatch. And so we studied the kinetics of flow using specific methods, and we developed the so-called undulation of the fibers, which allow the interfiber pathway to become much more homogeneous. And here is the demonstration of the blood and dialysate flow studied by what we call the mass transfer zone, which is the area where you achieve maximal utilization of the fiber down to minimal utilization of the fibers. We have an entire world of biomaterials applied to dialysis membranes. Here you see companies that have realized different type of membranes. And as you know, membrane characterization is basically by two main parameters. One is pore density, the number of pores per surface area. The other is pore size distribution. And this affects the membrane permeability coefficient in terms of diffusion, the ultrafiltration or hydraulic permeability, and the membrane sieving. So our attempt is to achieve a membrane which really resembles the glomerular basement membrane, somehow achieving maximum level of number of pores, but also regulating the size of these pores, because we don't want to lose albumin, for example, in the extracorporeal circuit. So we were among the first to publish about nanotechnology applied to dialysis. And as you know, in the industrial chemistry, there is this ternary diagram where you have a solvent, polymer, and non-solvent, whose application leads to the spinning technology and generation of membrane through specific polymer precipitation at the interface between solvent and non-solvent with different density that regulates this skin layer, which represents the real mass membrane separation layer. By doing this, we can regulate in part the number of pores, but also the mean distribution of pores, and we create specific curves of sieving which characterize different membranes in blood purification. But once you have done a good membrane, engineers are compelled not to just make one good membrane, but miles of good membrane, thousands of miles, millions of miles, because the consistency of the production of this membrane is a key issue. We can even design today the structure of the surface of the membrane with hydrophilic, hydrophobic, or microdomains. We can create a pore library using nanotechnology mechanisms, and we can even functionalize or surface modify the membrane. In this case, for example, you can covalently bind alpha tocopherol, allowing to generate a kind of antioxidant surface that blocks reactive oxygen species at the blood membrane interface where they are mostly generated. But membranes have always active components, and they tend to absorb materials such as, for example, endotoxin or cytokines. And speaking about absorption, we use in blood purification and we study sorbent materials that can be natural or synthetic. They have direct absorption or ion or cation exchange, immunoabsorption, and they are mostly done in granules that are structured in macro, micro, and mesoporous design. So this is a morphological analysis with electroscanning microscopy. We have done some collaboration also with the department of Professor Bagno, analyzing the structure of the external beads down to the maximum magnification of the pores. And here is a 3D reconstruction where it is evident that using a nanoscale molecular sieve control technology, manufacturers may adjust the pore size distribution. How to test this? We use absorption isotherms, which are not really easy to calculate. We mostly use the Freundlich and the Langmuir isotherms that are the best models that apply to liquids instead of gas for the solute separation. Of course, then we have to advise the manufacturer to design a cartridge that contains the sorbent. And in this case, you see that the flow phase, which is blood pathways, may require analysis of the interparticle, of the extraparticle, and intraparticle flow. And there are several variables that are regulated by these laws that you probably know much better than me. Including particle diameters, packing density, interparticle porosity, part tortuosity, length, diameter, fluid viscosity, and the Reynolds number. And we study the flow distribution utilizing medical devices such as the CT scan, where you can see the full utilization of the sorbent bed and the packet bed. We also analyze in a special experimental setting what we call the analysis of the mass transfer zone that allows us to calculate the distance between maximal utilization and minimal utilization of the sorbent that must be, for a well-designed cartridge, at least 1/3 of the length of the sorbent. We also use biohybrid artificial organs, but when you start using bioreactors, adding a biological component to a device, you introduce an enormous amount of variability. And this is one of the limitations of these devices. We also use mechanisms to decellularize the organs and then to inject a specific component at the different cellular level. For the heart, this is more simple because the heart is a rather nice but simple electrical pump. The kidney is much more kind of a biological and biochemical laboratory with different type of cells, and this makes our life a little bit complicated. We have designed in the past organs on a chip, and more recently we moved to 3D organ printing with the possibility to use bioinks in order to design 3D structure. We are a little bit far from the solution that may actually be something very important in space medicine, because as you know, when you design a mission to Mars, you are subject to an enormous amount of cosmic radiation, and for example, the kidneys, which are very rich on mitochondria, are the first organ that have a very accelerated senescence. And in the future, in order to go there, either we plan a specific protection or we will have to somehow readjust with a new organ the population that will visit Mars or more far away different stars. There is a kind of biology engineering convergence in molecular electronics, quantum computer, DNA replication, nanoscale networks, and this introduces a little bit the concept of artificial intelligence. I must tell you that preparing this talk, I was overwhelmed by the amount of information that I wanted to give in this lecture, and I had to make choice, and I had to surf and scratch the surface of several concepts rather than going into depth. However, how much artificial intelligence can help medicine? Enormously, enormously. And the most advanced question is, will computer ever develop consciousness? I spoke with Federico Fagin, who is the inventor of the microchip, and he is convinced that no, the computers will never develop consciousness. Well, believe me, after 50 years in science, I have no certainties at all. We have developed recently neural networks and computers based on organoids, which are aggregated of neurons. So how much these neurons will retain the property of the original owner of these cells, we don't really know. So the other thing is the application of the concept of quantum mechanics to the with the structure of new computers. As you know, computers work in a deterministic way, yes or no, 0, 1, but our brain works like by quantum leaps. And the truth is that if quantum mechanics is applied to computer, I cannot exclude creative evolution. And in doing this, believe me, you have to consider the light and the dark side of technology, because the light side is confirmative support, diagnostic algorithms, evidence-based guidelines, big data collection, data evaluation, fantastic. But the quality of data should be questioned. There is a deterministic approach still, no invention or creativity, one size fits all, which is not the case for our patient. Is this going to be a brain substitute? We say that artificial intelligence, and we prefer to call it, by the way, enhanced human intelligence for the moment, makes good doctors better, but bad doctors worse. I guess it's going to be the same for engineers. Now, how does all this fit into the career development of a biomedical engineer? So here I have a recipe which is totally personal on how I would like my bioengineer working with me to be. Now, you may start from the title. You say, I'm a biomedical engineer, which doesn't tell me anything, and you start from the top. Then you have to learn from the field experience and develop operation and managing solution. Or you may start from an individual experience and start from operations, learn with information, understand what unmet needs are, and therefore you can job positioning yourself with the leading process. So in the development of knowledge and skills, I see the basic level in which the engineer should know about different areas. But as you go to an advanced level, you must focus on some of these areas, and when you go to specialized and ultra-specialized level, you have to focus on at least one specific area, not forgetting your knowledge of the all different areas. So with the new information, in one sense, you have to surf all the knowledge available, but at the same time, you have to decide to dive into specific deep knowledge of one specific area. And so how should a successful bioengineer be according to my opinion, which is absolutely biased, you know? There is an initial stage, there is exposure to the medical field, there is understanding of the issue, and effective contribution to the field. Let's see what I mean for initial stage. The bioengineer should be curious and have some level of humility to understand that probably is more what he has to learn than what he has to teach. Willingness to explore the field, the human body, is something that is still 99% unknown. Commitment to excellence, you have to have a direction, and I think that this can only be achieved working in a team. You will hear many times this concept of teamwork. Exposure to medical field. The bioengineers that come to my department, I take them to make round. And every morning, medical brief, they should be there. They have to understand that any kind of solution they apply in the future is applied to real persons, human beings. And therefore, they have to have clear as a mission that the final ultimate goal is not to create a nice device, is to create a device that improves patient life. Identify unmet clinical needs only if you are at the bedside and you find a doctor that is struggling with something that is not working, you understand where your competence can actually help. And then you can make your research agenda. Now, understanding the issues, of course, include a compelling evidence for safety and regulatory aspects of devices. I always discuss with engineers, they said, this is a perfect membrane. Yes, it is a perfect membrane when you use a saline solution. But when you use blood, the protein clogs the pores and the membrane doesn't work. So why is this stupid blood not working like a saline? Because blood is a non-Newtonian fluid. The relationship between shear stress and shear rate is not so regular. It's probably more like a pseudoplastic fluid. It is a fluid that has a very strange behavior. When you run the blood at high speed, viscosity decrease. When the blood is sitting there, viscosity increase. So it's a tremendous fluid, but this is what we need to work on. And interacting with nurses, especially in the team, makes the bioengineer a real person that understands what the issues are. Now, I want to mention that also when you design something, you have specific challenges. One is biology, but the other is the human nature. Just to explain to you, we did a machine safety analysis, and on more than 1,000 treatments worldwide, we analyzed the number of complaints for machine malfunction. Well, 84% derived from machine misuse, which means something. It means that nurses and doctors are very creative in not utilizing the devices in the proper way, but maybe your instructions were not so good, or simply the fact that there was no enough training. So documented failure was only 16%, and therefore you have to deal with human nature. To my collaborators, I always say, doesn't matter what you say to another person. What matters is what the other person understands. Now, finally, you can effectively contribute to the field and find your role in the team. Share your knowledge, use imagination and creativity, and for this, Italian engineers are very, very good, and propose new ideas and solutions. But the success is based on teamwork. Working together is the real result. And I want to mention something. When I was young, I was reading science fiction, and I advise you read science fiction, because what is science fiction today is science tomorrow. There was this starship that was including a large number of scientists plus one. And this scientist was expert in nothing except in putting together the different scientists. It was called the connectivist. Now, the connective tissue in the body is what keeps all the organs working together, right? So I wrote a book entitled The Connectivist, because I wanted to explain that the multidisciplinary approach is really, really the answer to a successful project. And we created in our institute what we call the round table of science, with the medicine in space, bioinformatics, prosthetics, artificial organs. Why is it a round table? Round table because there is nobody at the head of the table. Everybody can give a contribution to the final project. And this is our institute that includes several of the different disciplines, including ethics, because, my friends, technology is a very fast developing discipline, but it has to be followed and mitigated by ethical consideration. We use the term green to define several missions like genetics, robotics, e-health, eco-compatibility, and nanosciences. And this is our bioengineering laboratory in our department. Now, let me tell you a story, because if you don't make examples, probably you don't understand what the feeling is. In Okay, the story requires a little bit of advancement. Can you do by hand? Okay, let's see. No, suddenly this doesn't work anymore. Anyway, can you go ahead? So our mission is to change the future of our patients, right? At this point, in 1946, this guy, Professor Alwell, entered the room, the sitting room, fireplace. His wife was reading a book. He said, "Honey, do you think blood can be washed?" And his wife said, "As far as I know, everything can be washed." And this was the beginning of dialysis. After that, we developed machines and made dialysis available for everyone, but patients had to be dialyzed for 13 hours, 3 times a week. I mean, this is slavery, is not a cure for a disease, right? But after that, and after many evolutions of this machine, I wanted really to put all the entire evolution, but I had to come just to the last generation of machines. Basically, through online monitoring and the biofeedback that makes the machine interacting with the patient and leading to an automatic response in presence of specific signals, we ended up with the profile of the therapy guided by a biofeedback system. And today, patients do 4 hours, 3 times a week or less. We changed, actually, the life of these patients. And we're working more on this through this multidisciplinary approach And we are in Padova, the Galilean method, right? Now, Galileo changed the current paradigms and imagined the future. He observed the problem and seeked the solution, and he used the new experimental scientific method. He was obsessed by dimensions. Not many know that, for sure, he invented the telescope, but he also invented the microscope. He was obsessed by this concept of dimension. And if you look at the evolution of pacemakers, again, the heart, stupid electrical pump, is governed by now a pacemaker which is less than a coin. Look at dialysis machine. Up to the last generation, they still look like a big piece of furniture, right? So our idea started to try to miniaturize the machine and put it on the patient, creating what we call the wearable artificial kidney. Because miniaturization means portability, wearability, implantability, mobility, dehospitalization, home-based therapy, impact on welfare, costs. Now, I had the pleasure to meet Galileo Galilei in the university rectorate where there is a chair of Galileo Galilei. But in real life, I met Richard Feynman and Eric Drexler, two giants who made significant contribution. Feynman received the Nobel Prize, and Drexler was crazy like a horse, but he developed the concept of nanotechnology. Now, going back to Galileo, he mentioned the square-cube law, in which basically he says that machines of different dimensions may require specific actuators, system of measure, and calibration. And the square cube applies to physics, biology, even to avionics. Now, simple objects can still work if you change the dimension, but complex machines or biological systems may dysfunction when dimensions are not maintained within tight ranges. Let me make an example. Why do you use a pediatric stethoscope? Not because the kid is small, but because the frequencies you can identify with the different membrane size are different, and therefore you can feel the murmur of specific defects in the heart of the small patients. Why are cells not getting bigger and bigger? Because they need a specific surface to volume ratio to transfer nutrients and waste products, and therefore they prefer to divide. So even complex machines like cells require a specific dimension. For this reason, we took the lessons from Feynman and Drexler, and we tried to take advantage of new sciences, microfluidics, nanotechnology, microelectronics, to design the concept of wearable artificial kidney, which at this point, as you have understood, could not be simply a miniaturization of a large machine, should be a new kind of project. And we developed conceptual frameworks, technology dedicated, ergonomics and design, energy sources, management and logistic plans to develop this. And this was the first patient ever in the world in our hospital walking with the dialysis machine on the belt. And this is a young lady wearing the wearable artificial kidney near her mother. They have a genetic disease, and the mother is dialyzing with a classic device. So this is a new evolution of the wearable artificial kidney. We are trying to make it a little more wearable, and different designs are now utilized in the world until we plan to be able possibly to generate an implantable device. Now, shoot for the moon. Even if you miss it, you will land among the stars. What does this mean? We said, okay, who is the guy who will really benefit from miniaturization? The baby. The baby is, the neonate is the patient that will really mostly benefit from miniaturization. So neonates with renal insufficiency present high risk of morbidity and mortality, and they have an additional risk due to lack of dedicated technology. The technological transfer between adult and neonates is lacking, is missing. And in practice, this makes disease for the neonates like orphan disease. So we have peritoneal dialysis, we have hemodialysis, but they are not really applicable in the neonates because you need to have the right tool. Look, the smallest circuit in an adjusted machine for neonates, but the machine is originally for adults, is 95 milliliters, which is almost 1/3 of the blood volume of the entire neonate. This cannot work. And in fact, if you want to do a special task, you need a special tool. You cannot think to fix a watch with this tool, right? So the concept was that we wanted to have at least less than 10% of the circulating blood volume in the neonate. And smaller patients require blood priming that fulfill this approach. So the idea of miniaturizing a machine from the adult was simply not working because the neonate is not a small adult. It's an entity completely different. So we had to start a new project. We wanted the blood flow between 0 and 50 ml per minute, an ultrafiltration rate between 0 and 5, an accuracy of 0.1 milliliters per minute, and a priming volume less than 30 milliliters. And we started with an operation of crowdfunding, team building, charity events, project design. Here you see that what Professor Bagno said before, we did it with specific hockey games to collect money. Here I'm playing with the Nomady, as you can see here. And finally, we collected 380,000 euros. A project like this in the United States with a simple venture capital round would have collected 50 millions. But in Italy, that was the amount of money we collected, totally independent, sufficient to develop our project. We started with prototypes, and here you see different devices trying to match the size of the patient with the size of the device, which was small enough but still not very well designed. And here you see that I'm not a great designer. Certainly this is not an AutoCAD, but this is my first drawing on how I wanted a machine. So I went to 2 engineers and I said, this is what I want. Don't bother me if this is not a good design. You will make a good design. And in fact, they did it. They started with specific 3D design, and finally, in 2010, thanks to these 2 nice engineers, that I have to tell you, we were working undercover because the companies of these guys didn't want to develop this device because there is no return of investment when you do a machine for neonates, too little numbers, right? But these engineers were kind enough to help me to develop this first prototype of the first world design machine for dialysis in the neonate, with several new features, including rotor and stator with specific characteristics, mini filters, a scale with 1 gram sensibility, and a specific one-button operation system with a lot of electronics inside, but a lot of simplicity outside. We tested in the labs. This is a Korean bioengineer now in the Institute of North Carolina, and this is a colleague from Bambino Gesù Hospital in Rome. And we tested this, and finally we came up with the final machine, which was the result of the collaboration between a bioengineer and a doctor. And every single component was different. And in July 2013, it was approved for human use. Not to tell you, 1 month later, to our hospital comes a baby with a tremendous hemorrhagic shock after delivery. And here you see the enormous amount of blood collected in this area. The baby was extremely fluid overloaded, and she had acute kidney injury and acute liver failure. We placed in the femoral vein a double lumen catheter, which was French, 4 French bilumen. 4 French bilumen means like a hair. It was a catheter that was never utilized before because machines could not tolerate the pressure drops in the system. And this picture is the first picture of the first neonate ever treated with the carpe diem. And here you understand immediately what is, with the same type of catheter, the jumps in pressure due to a typical rotary pump, a peristaltic pump for adult machines, and what was the pressure condition due to the small rotary pump in the carpe diem machine. The baby was treated for 25 days, not without complications. She turned yellow at a certain point, so we had also to use with the system single-pass albumin dialysis and even plasma exchange. But the machine was designed for this, right? And so after a series of treatment, you see bilirubin going down, liver getting better, creatinine going down, and starting to decrease the severity of acute kidney injury. And finally, the baby was extubated after 25 of extracorporeal therapy. Now, it is interesting that we had a chat with the Cincinnati Children's Hospital, which is a friend of mine, Dr. Stuart Goldstein. At the beginning, he said, we got a French Belumen 4. You aren't going anywhere. I said, it's working, constant flow. It is impossible. The baby is surviving. It is a miracle. Finally, I said, the baby recovered. He said, this will change the way we will do medicine in the neonate with acute kidney injury. And there is a book telling this story. And the entire world was interested into this, and we published in Lancet. For 1 month, this story was on the top page of Lancet, which is a nice journal for us. And you see that there was the picture I showed you before. And this is the baby after she recovered. And I can tell you that this smile is really worth 45 years of medicine and a fantastic cooperation with bioengineers and with friends. Now, if you look now at the European Pediatric Registry, mortality in this case has changed from 82% to 29%. So we really change their future. And that now is FDA approved and utilized all over the world. We can say somehow that this is the heritage of Galileo, right? But this is not finished. We're now moving to smaller and more efficient devices. Here you see the last 81 project that we are developing. So my suggestion to you is be curious, be critical, be intelligent, think outside the box, or even get rid of the box. Try to be creative. I would like to also suggest to live with passion. Be passionate because you will lift the world. All the skeptics will let it drop. And finally, one last recommendation. Cultivate the humanistic spirit of science because ethics and science go together. Be the connectivist of your team. Be one of the members of the round table of science. And by doing this, sometime use less internet and a little more of Cabernet. Thank you very much.
Thank you, Professor Claudio Ronco, for your very interesting lecture. That's now open for discussion. Are there questions for Professor Ronco?
What brand of Cabernet?
I would like to ask you your opinion. You touched the number of different items. And in particular, you talk about artificial organs, both with regard to mechanical devices or bioengineered devices. In your opinion, which will be the more convenient direction for research, going through the mechanical devices, trying to miniaturize them, or try to develop something really biological?
Thank you. If I could answer this question, I would probably win the Nobel Prize, and I will also
It could be a chance for you, Claudio.
millions of dollars. But I think you have to start from different configurations in your research. First of all, when you use inert biomaterials, and inert means that they have to be at least biocompatible. Actually, even better, they have to be hemocompatible. I think that you can design a completely standardized, reproducible device, because production may become very well standardized. When you introduce a biological variable, at that point, you introduce an enormous range of variabilities. Nevertheless, you know today that what we call the biological drugs are drugs that we develop asking bacteria to print a specific protein sequence, which is then used to develop the drug. So it can be done. We know that today we have not enough knowledge, however, and possibly in my life I will not see an engineered biological kidney implanted. But still, we shouldn't stop, and we should move towards specific research projects. This is the role of academy. Unfortunately, this cannot be the role of industry, because industry asks for a quite rapid return of investment. And this makes the difference in research between evolution and revolution. Evolution is a kind of progressive, small improvement and implementation of new technology of an existing device. Revolution is something that everybody thinks is impossible until somebody that doesn't know it come and invent it. So I think we have to move in both directions. First, because we can significantly improve our mechanical device, our biomaterials, our artificial organs. On the other hand, on the other side, we have to explore avenues that include the study of biology. There is another issue, which is many of these technologies, for example, rely on large amount of water. Large amount of water is not available everywhere in this planet. So the other effort should be, rather than creating the new rocket science, to make these devices available for everyone who needs it in this planet. Believe me, today is not like this. So there are several different areas where we can work, including the fact that we have to be environmentally friendly. We have to study the possibility to generate systems to regenerate, to recirculate, to reuse materials. The spectrum of research that you have in front of you is enormous. Believe me, do not listen to somebody, especially if you're young, that come to you and say, oh, this has been done already, oh, this has failed. Please, tell them to, don't tell anything. Do not accept the idea that something cannot be done. It can be done. Maybe not now, maybe not tomorrow. But remember, is the question I make to my students when I start the academic year is, do you believe that somebody in this room will win the Nobel Prize? And they look at you a little bit, and I say, why not? Someone has to win the Nobel Prize, right? It may be you. Now, it's important not to leave the frustration of all your life if you don't win it. And it will be extremely difficult. But remember, it may be possible, and you guys can change the future of our patients. We did it. We did it through a teamwork. You can do that. Now, I use all these nice words to escape the question of Professor Bagno, you understand that. But the reason is that I don't want to focus on one specific field of research. I think we have to be broad, and we have to accept the idea of this bionic convergence with engineering and biology that more and more in the future, just think about DNA-based computers, quantum computer, and et cetera, et cetera. Quantum physics is just at the beginning. We are just starting to understand something. And the moment we understand something, quantum physics changes completely, right? So this may actually revolutionize the future. But be careful, because when you have your bionic convergence, you may arrive to a point, and I don't want to mention Skynet and Terminator, but I want to mention that the interaction with machines will be more and more complex, whether or not they will develop consciousness. And that's why sometimes science has to be slow, because ethics should accompany the development of new technologies. This is what I think. I have always been a great fan of technology. Artificial intelligence is the first technology that really scares me. First, because I think it's successful because natural intelligence is lacking a lot. But second, because you cannot really have a governing body of this. And since the world is full of idiots, somebody one day come up with applications and use of artificial intelligence we are not what this was designed for. There are other implications. I just finished to write a small editorial saying that we will be surely utilizing artificial intelligence, which is derived from a combined biological and electronical research, but this will help us for guidelines. An artificial intelligence system can look at millions of chest X-ray. One radiologist can look at 100,000. So the experience of big data and interpretation provided by the artificial intelligence would be important, enormous. But at the end, the problem is that this may guide our behavior, may guide our decision, and I didn't project one slide because I had to cut down some slides. I also asked ChatGPT, who is Claudio Ronco? The answer was, he's a famous bioengineer who developed artificial organs. Completely made up answer. But then I said, are you sure? Answer, well, looking a little bit better, he's a medical doctor, but he looks like a bioengineer. That's strange. So the system learns. And when it learns from existing experiences. But in the future, who will give new experiences to the system? And at the end, the editorial ends with this concept of quis custodiet custodes. I know that many of you probably have done not Latin at school, but it's a major lack, believe me. It's a good idea to study some Latin because the ancient people were not so stupid. And today, quis custodiet custodes is a very important question to make for the future of artificial intelligence.
Thank you, Claudio, for your answer. Are there any other questions? Please.
Hi, Marco Puenassi, University of Trieste. You were telling about, okay, the beneficial of the patient. I work a little bit with implantable device and wearables and interfaces. And what I see marvelous device with marvelous functionalities, more or less hidden behind like hiders, like worst chassis, worst device by itself. The command of my garage is better to some device that you implant in the patient and you use the interface that you give to the patient. So my question is, for you, how important are the non-functional parameters of the device? So how beautiful they must be, how the style, the usability is important in the development of this device as a medical doctor and also bioengineer, like ChatGPT said.
Well, you know, I wrote a book entitled Aesthetics and Medicine. I am in favor of thinking that the beauty is a cure. Patients love to stay in nice environment, well-designed hospital, etc. In terms of artificial organ and devices, having a very well-designed with high ergonomy, high capacity of interaction is a key issue for the success. Now, if you take the phone, right, and I can tell you from this phone that this camera that have like 1 millimeter surface on top of this, Steve Jobs would have killed the people because it's not so smooth and not so nice. But this keeps all the complexity inside and all the facility of interaction outside. So I think that the device must be easy to use, light, cheap, friendly, especially if you have to wear it, not to think if you have to implant it. So that component is at least as important as the functional spectrum. You have seen our last project, the AD1. Oh, sorry, I made it the wrong. We designed this small device for congestive heart failure. I fight it with the engineers because I wanted like a cigarette package. And for different reasons, including batteries, etc., they had made this a little bit bigger. But I continue to fight because the smaller and the lighter it is and the easier to use, the patient will certainly use it. And there is another component that are nurses. In machines, if nurse love that machine, the machine works. If nurses hate that machine, it will never work. Because at the first malfunction, instead of troubleshoot, they say, it's not working, and they take it out. So absolutely, you have to work on the interface, you have to work on the characteristics, and at the end, you might have seen our first wearable belt. It was very ugly, but we used off-the-shelf materials. Then you have to design specific materials. Today, with the 3D printing, is much easier. In the past, you had to do molds, etc. But this requires investments. And these investments can hardly be done by the academic world. You can do it in conjunction with companies. But at that point, you have to accept that once they develop something, they want the span life of this product to be at least 5 years, while at the first year, you would be ready to have generation 2.0. That's life.
Thank you.
Thank you, Professor Ronco. We are just 3 minutes late, so I know that now the first poster session will be open, so On behalf of the National Community of Bioengineers, I would like to thank you so much for your lecture. Thank you, Professor Ronco, and good afternoon to everybody.
Thank you.